In this video, we're going to take a look at the 5 different classes of antibodies and what occurs during the primary and secondary immune responses as we encounter an antigen. So each of the classes of antibodies, or immunoglobulins, is identified by the particular amino acid sequence and then the shape of the constant region of the immunoglobulin, also called an Ig. And there are 5 classes of immunoglobulins: IgG, those are the gamma globulins; IgA, alpha globulins; IgM, IgE, and IgD. So those are the names of the 5 classes. And so we'll look at each one of these 5 classes. So we'll start with the IgG, the immunoglobulin G. These are the most abundant class of antibodies found in the blood, and they can be as much as 20% of plasma proteins, especially when one is in the midst of an immune response. And they're produced in the largest amount during a secondary immune response. And just as a reminder, recall that this portion here of the antibody, that's the constant region, and so it's the sequence of amino acids there that determines that this is an IgG antibody. So the antigen binding sites, that's this region here, the variable region, they attach to antigens on microbes, and the tissue binding site, the constant region here, attaches to receptors that are found on phagocytes. And so once the IgG antibodies have bound, then phagocytes can bind to them and then phagocytize it. There's also regions of this antibody that act as complement binding sites. And so that allows the first of the complement proteins to bind and initiate that whole cascade of complement protein formation and all the effects that we see of complement on immunity. So IgG are able to cross the placenta, and they form the major protection of the developing embryo and fetus from infection when, if the mother were to encounter an antigen of some sort. And it's also found in breast milk. So as the infant nurses, then it also provides protection to the infant as well. Okay, so that's IgG. The next one we'll look at is IgA. IgA functions to guard all the entrances into the body from bacterial invasion. So we find it in the mucous membranes especially. And it works by binding antigens and then activating complement. So there's only a small amount of IgA in blood, but it's much more abundant in body secretions. So we find it in mucus, in tears, and it's also found in milk and saliva as well. And it attaches into the mucous membranes that line the digestive tract, respiratory tracts, urinary, and reproductive systems as well. In blood, it can be found as like one individual antibody, but it can also be found when the antibodies bind together as dimers. That means 2 of them. Trimers, sometimes they're bound together as like a threesome, or as tetramers when they bind together in groups of 4. In our body secretions, it's found as a dimer, and it can, in that dimer form, so when it's found as 2 of them bound together, it protects it from being destroyed by enzymes, proteolytic enzymes that would normally break down proteins. And it turns out that if a person is genetically unable to produce this dimer form, then what we see is that they have a higher incidence of dental caries. The cavities are more likely because their saliva doesn't protect them from bacteria that can be found in the mouth. The IgA doesn't cross the placenta, but it is very abundant in colostrum. That's the first secretions that come from the breast following pregnancy before the breast converts or shifts from making colostrum to making milk. So for the first day and a half to 3 days, something like that, rather than producing milk, the breast produces colostrum, and the colostrum is filled with IgA. So again, providing some protection to the infant once he or she is born. The next class of immunoglobulin, I suddenly can't say it this morning, immunoglobulins is IgM. IgM is the first antibody that's secreted into the blood as part of the primary immune response. On B cells, as the receptor for antigens on B cells, it is found as a monomer, a single one of those antibodies, but it's secreted as a pentamer. This is a pentamer here, which is a 5 antibody cluster of antibodies. And it's called the M form, as M for macromolecule, by the plasma cells. So as plasma cells are making it, they bind them all together into these 5 antibody clusters. And they're connected by their heavy chains and by what's called a J chain. So here you can see the heavy chain, the purple chain, is connecting them. They're connected in this way. And then there's a J chain that closes it together. And so because each antibody has 2 binding sites for antigens, these IgM have a total of 10 binding sites for antigens. And this causes clumping of antigens when they bind into a great big molecule that is very easy for complement proteins to find and then to bind to. So those complement proteins do a really good job of finding these just because they're so large. This is the very first antibody that's produced by the developing fetus, and they are the antibodies that are responsible for ABO blood group types. So, so when we think about ABO, it's IgM that we're talking about that's found on the surface, that's produced. So if you think about anti-A antibody, it's this big pentamer that we make when we're making anti-A antibodies or anti-B antibodies. They are too large to cross the placenta, but they can be found in high levels in blood, and that when they are in high levels in blood, then that indicates recent primary exposure. So that's one way that we know that someone has been recently had a primary response, because they're such a large part of that primary response. Because again, if you remember, IgM is the first of the antibodies that's produced during a primary immune response. Okay, so that's IgM. IgE is produced and then released into body fluids. The tissue binding site will attach basophils. So our constant region here can attach basophils and to mast cells. And of course, the variable region will attach to the antigen itself. It's very common in tissue fluids, but it's not seen very often in blood. So the antigen's binding sites then are available to attach to the antigens that it finds, so drugs, and it's found pollen, some foods, and these are antigens that act as allergens that trigger allergic responses. Because remember, IgE is very active in allergic responses. So as so if we have, say, here's a mast cell and it's got IgE here in its, as part of its membrane, then when the antigen binds, to the antigen binding regions, then the basophil, or the mast cell that's holding it, produces lots and lots of histamines and other inflammation promoting chemicals, and that's what we see as the allergic response, is all that histamine being released. And IgE is especially abundant in patients that have allergies, or when we see the extracellular parasites, like worm parasites. Those guys will stimulate the production of IgE. And then the last group is IgD. They're found mainly on the B cell membranes. They're rarely secreted. And while they can bind antigens, we don't know a lot about what their function is. But we do know that it's associated with immune disorders or autoimmune disorders when we develop immunity against self cells, because some of those autoimmune disorders are associated with an increase in IgD levels. So stay tuned. There's more to be understood about what IgD does, how it operates. Okay, so the primary immune response is the immune response that occurs the very first time that a person's body encounters an antigen. So a host B cell finds an antigen, and when it recognizes it, then the B cell divides, forming plasma cells that start to produce antibodies. And of course, helper T cells are involved in there, but we're just thinking right now about the production of antibodies. So within about a week, IgM begins to appear into blood. Recall that that's the pentamer, the five-part cluster. And over time, so here, within about a week, we see this peak here in IgM, if you're looking at this graph here at the top. But over time, there's a shift, a decrease in the production of IgM as that goes down, and then IgG picks up. And so here we see this peak in IgG that occurs sometime at about 20 days, give or take. And it's that IgG peak that generally stimulates healing from the antigen. And as antigen then is eliminated from the body thanks to the work of the IgM and IgG antibodies, then antibody production declines, and antibodies can become virtually undetectable in body fluids. Okay, so here's the exposure. Let's just make sure we're understanding this graph. Here's exposure. Not much is happening in the way of antibody production because we got to get plasma cells and helper T cells all activated to the same antigen. Then we see an increase in IgM. And then over time, IgG picks up, so we get a total antibody response, and then in time, it wanes and declines, and there's not much antibody being produced. And here we see, the B cells begin to produce IgM, and then in time, they begin to produce IgG, and memory cells are made. Okay, so those memory cells are going to be important then through to stimulate the secondary immune response. So populations of these non-dividing memory B cells can persist in the lymphoid tissues. They can be there for months, years, and sometimes even for the entire life of the individual, waiting to find that antigen again. If they find that antigen again later on, so here we see there's a break, time passes, then the memory B cells will discover that antigen because they're going to be in much greater abundance than the ancestor of that original cell. So as those memory B cells encounter the antigen, then they will divide, and so here's the memory B cells. You can see them dividing, and some of them are going to produce plasma cells. So here we see, once again, there's IgM produced first, and then IgG produced second, but we produce, again, a whole population of memory cells as well. So as those plasma cells start producing IgM, we see a response, but what we see is that there is a huge response in IgG. So overall, the response is much greater in the secondary response. And also take a look at the time scale, where it took up to 15 days or so to hit maximum antibody production, and maximum IgG production was out here at like 20 days. Notice we get to maximum antibody production much sooner. And that rapid response and the very large response is just overwhelming. And because of that, we prevent the disease generally because of that rapid, overwhelming production of antibodies. Okay, there you have it. The classes of immunoglobulins and the primary and secondary response. You know what to do if you've got any questions. Oops, I'm sorry. One last thing. Two last things, actually. So antigens determine what mechanism is used to sensitize B cells. T-independent antigens, these are antigens that bind the B cells and activate them and cause direct proliferation of B cells as they form plasma cells. They're T-independent because no helper T cell is required to get them going. And so in the process, we don't form any memory B cells, and we only produce IgM. So these T-independent antigens, they produce no memory. And the kinds of antigens that stimulate this response are generally the polymers, molecules that are built of repeated subunits. So things like the capsule polysaccharides or that lipopolysaccharide, remember endotoxin, that's found in the membranes of the gram-negative bacteria. Also flagellin, which is the protein that's used to build the flagella of a bacterium, and the double-stranded RNA that's found in some viruses. So they are T-independent, and they don't produce memory or IgG antibodies. Most antigens are T-dependent antigens, and when the B cell encounters that antigen, it's sensitized to it, but the B cell becomes an antigen-presenting cell. It makes contact with a helper T cell that's been sensitized to that same antigen, and it activates it and causes it to secrete lymphokines. Pronunciation is rough this morning, I tell you. So those lymphokines then are secreted all over the B cell, and the B cell then is stimulated to proliferate and differentiate. It produces memory B cells and plasma cells, and these are the ones that produce the kind of primary response or secondary response that we just talked about, in which the plasma cells start by making IgM, but over time a group of plasma cells begin making the IgG antibodies as well, and we get memory from this. Okay, now that's the end of the lecture. Again, you know what to do.